Hello, everybody. Welcome to Natural Growing Through Biology. I'm Dennis.
I'm Steve.
Today, our podcast is going to be abiotic stress, biology as the buffer. So we
it
got a lot
always
to talk about.
it always is. It's so important, but we're gonna start off on a happy note. No, womp womp. Doom and gloom. The reality of where we are. Since we're talking about abiotic stress, abiotic are non-living stresses. So oftentimes this is just referred to as the environment. And when we look around the environment, we see some strange things going on. Soil degradation, both global and US. We're seeing dust storms, heavy precipitation. Lake Tulare for the first time since 1898 here a year ago.
Well, yeah, and actually, it's funny on Lake Tulare, it was actually drained because of they diverted the waterways for agriculture and municipal uses. And that's why Lake Tulare dried up.
Yeah.
It was actually one of the largest lakes
Yeah.
west of the Mississippi, which is, I mean, pretty unbelievable. And it's had a few over the years times where it has had water in it. But in 2023, I think in 93, it had water in it. And then like in 97, but 2023 was the first time. I mean, it flooded. It took out farms.
It was a lake
unbelievable. A huge lake.
Yeah.
Yeah.
So the temperature shifts and changes, and we need to keep in mind as temperature goes up, warm air holds more moisture. So your stressed plants are required to go through more evapotranspiration to cool themselves, meaning we're extracting more moisture. The plants need more moisture to stay cool. They need more moisture. And through that evapotranspiration, they're burning through more moisture. So as the warmer the air gets, the more water those plants need.
You know, and we even see now it is a shift in frost-free days, the
Yeah.
number of frost-free days. You know, on the West Coast, basically, we're seeing tonight 16 to 19 fewer frost-free days. Now, the amazing part about this is when we start to talk about cherries or apples or anything like that.
Early flowering.
Early flowering, think about it. We're flowering 20 days earlier than we did before, but our last chance for a hard freeze has not changed.
Nope.
So we just have a longer period of time where we are subject to Mother Nature.
Roll those dice.
Yeah. So everything that we can do based off of overall plant health is what we're going to talk about today.
Yeah. And it all boils down to, and the term is commonly referred to as climate or weather weirdness. We're seeing weird things happening. I mean, the tornadoes we've been having, it seems like we've had a very heavy tornado season already this year. Who knows what the hurricane season is going to be like? We're seeing more and more of these weather events. So we need to keep in mind, well, we can move around. Plants cannot. According to Ian Baldwin, Max Planck Institute, plants' genome has probably an order of magnitude more genes involved in environmental perception than most animals do. Most plants have to. They have to really tune their physiology and biochemistry to what is going on, and they need a very sophisticated system of perception and response. Communication. There has to be communication. He's focusing in this research primarily talking about the plant's own genome gives it a great deal of plasticity. It can move, it can shift, it can work, but only to a certain extent. And that's kind of where we're bumping up into some of these issues. We're changing the environment around these plants. And we see as we change the environment, what do we say regularly?
The environment dictates expression.
Yeah, so we're seeing shifts in weed species. We're seeing shifts in plant species. And we need to use those as an indicator. And it's not just temperature shifts that we're seeing. We're seeing moisture shifts that lead to different weed species, salt shifts that lead to different weed species. But we need to keep in mind that's the plant side, and that's a good portion of the genome, but that's ignoring the rest of the plant's genome. The microbiome is critical, the phytobiome, all those organisms that are on, in, and around, or at least should be on and around the plant. And those are what we often say, that biology is functioning as a buffer. It's helping protect the plant. It's also communicating with the plant. It's utilizing a wide variety of different molecules to talk to the plant, to talk to each other.
Yeah, and you know, that's part of the thing. Like you say, the plants can't move. So we have to do everything we can soil-wise, digestive system-wise, biology-wise, nutrition-wise. But sometimes we get so focused on the biology making nutrition available to the plant, we forget all those other things that the biology does and how it communicates with the plant and the things that the plant can actually do. And that's what we really want to talk about here today, is kind of dig in to all of these different things, which we refer to it as biology as a buffer.
Yeah.
But let's talk what those buffers really are.
Yeah. So one of the things that we talk, okay, so as you add weirdness, you add stress, because that plant, that temperature, that shift, all of those are going to force that plant to respond. Every time a plant has to respond, it takes energy. It takes energy to upregulate certain genes, downregulate other genes. And if it does it at the wrong time, at the wrong rate, that's just wasted energy. That is just dumping energy. That means we have loss. Our genetic potential, every time we add a stress, we lose yield. I think about it as like a water balloon. You have a big water balloon filled. Every one of those stressors is like poking it with a little needle, and you see those little streams flowing out. At the end of the season, maybe you've got a mostly full water balloon, or maybe you've got a shriveled little weird looking raisin thing.
Yeah. And that's what it all comes down to, yield. So let's talk about what things these biological communities and plants are doing as designed when we let them function as designed. Again, environment dictates expression.
Yeah. And so research is showing that when a plant is exposed to stresses without a non-PGPR, and this research was done non-PGPR versus PGPR, plant growth promoting rhizobacteria. And when the researchers are measuring this, they see when you add a stress, whatever stress that may be, you see increases in abscisic acid, ethylene, and reactive oxygen species, and a decrease in nutrient uptake when we don't have those organisms present. By then simply adding those plant growth promoting rhizobacteria to the root and allowing that communication, allowing that perception to happen, they see a decrease in abscisic acid, ethylene, reactive oxygen species, and an uptake in nutrients. So that's what we want to see. We see a significant shift and change in that plant's hormone and profile of those hormones, allowing it to grow more normally, because that's what we want.
Yeah, and we often talk about basically how the plant is able to express itself, how it is able to control itself in that environment. And the nutrient uptake is not just directly correlated to the fertilizer that we put out there or the microbial digestion. A lot of it has to do with also how is that plant functioning? How is it able to take up that nutrient? What we talk all the time about excesses. What excesses are blocking that plant from uptaking nutrition? And as we see here, nutrient uptake goes up when these guys are available and present. We realize that.
Rhizophagy.
Yeah, but there's a lot more going on.
There is, and that's a good reminder. I mean, when we talk about reactive oxygen species, a plant is generating reactive oxygen species all the time. It's utilizing those to help defend itself. It also uses that to help break down molecules, break material down. It takes a lot of trace minerals, copper, iron, zinc, manganese, to create the enzymes necessary for breaking those reactive, or basically satisfying that reactive oxygen species. You need antioxidants. So the plant has to generate a higher level of antioxidants. Many of those are some of the plant's metabolites, some of the other molecules the plant likes to have in abundance. Those microbes are helping with those molecules. They're helping with the uptake of those trace minerals that allow the enzyme pathways to function. So we see a lot of those nutritional changes and those hormone changes. A big area that we like to talk about, and something that we talk about pretty regularly, is that rhizosheath, that rhizosome, this zone on and around the root. And when we go out to the field and we dig up a plant, and we can't really see any root, there's no little white stringy things down there. It's all this brown material that's globbed on and around the root, like imagine a leather glove on your hand. That's good. We like seeing that, don't we?
Oh, we love to see those dreadlocks around that root of what we talk about. And so often, we chat all the time about these are microbial communities that are living on that rhizosphere, and they're helping the plant with all of these different functions. Basically, we talk about pH, but some of the stuff we don't talk about is some of the I call it scientific, or digging in a little bit deeper of actually what they are doing based off of their function, not only based off of nutrition and producing soil health, but other functions that they have in and around that root structure when it comes to developing overall plant health.
Yeah, no, they're purposefully engineering the soil system and soil environment around and on that root. All of those little microscopic soil particles that are being bound together and then surrounding that root like a glove Or like insulation. I really like to think about it like insulation. It's a buffer. It's creating that zone that separates the plant root from the bulk soil around it. And the microbes are doing that with extracellular polysaccharides. So these microbes are generating a lot of glue. EPSs are glue. It's the biofilm. It's that stuff you wake up in the morning, you got slime on your teeth. That's biofilm. That is microbes purposefully working together, creating an environment that is better for their growth. In a soil environment around that rhizosheath, all these microbes are taking the little microscopic root particles, the microscopic bits of humus, the microscopic chunks of sand, clay. They're taking all of that and binding it together around the root. That's what we love to see, because now we have created a buffer zone. And that buffer zone is going to contain a higher level of organic matter. We've pumped more microbial biomass. We've pumped more exudate. We've created a zone that has a better moisture holding capacity simply because it has more carbon. Carbon is critical. Everything is revolving around carbon because we're all carbon-based organisms. That's the thing that these microbes need. It's an exchange. It's a back and forth. Nobody's getting away with a job for free here. The microbes are extracting nutrition and they're feeding it to the plant. In exchange, the plant uses some of its photosynthetic exudates, its sugars, to feed those microbes so that they can continue to do their jobs. And whether we're talking about sodium, heat, low temperatures, droughts, heavy metals, this extracellular polysaccharide zone is creating a protective sheath, a protective buffer, a protective insulation around that plant root. And we see some pretty interesting things going on in that environment. We see temperature shifts, both positive and negative. You've seen some pretty cool things out in the field, haven't you?
Yeah, it's funny, like you say, excessive temperatures or actually increasing temperatures around that root area in order for that to be more accommodating to the microbes and also to the plant that is functionally growing within that soil
They
environment.
want a happy home.
Yeah, and I remember a few years ago, a golf course superintendent in Colorado called me and he said I needed to come to his course. And I'm like, I'm thinking, what's going on here? This is March, there's snow on the ground. And I drove out to the course and it was amazing because you looked across his entire golf course, which was solid white.
Yep.
And his 18 greens, which are on a biological program, were all green and had no snow on them.
Oh, I wish we had a drone back then.
Yeah, because that biology was creating a different environment, a little warmer soil temperature.
What happens in a compost pile?
It heats up.
Yep.
And that's what we're doing.
Yeah, the natural digestion process generates water, generates heat. Sorry, go ahead.
No, and and I tell this all the time. A lot of the courses, they love the fact that in Colorado, everything around them would be brown and dormant. All the bluegrass and the greens that were on the biological program would be lush and green because they had a warmer temperature. And really, when you start to talk about yield, the greens are the golf course equivalent to yield. They had a very high yield because, you have a nice warm day in March and people wanted to get out and play golf on these beautiful lush greens when you had a nice sunny warm day.
Oh, and now that plant, because it's green, it's photosynthesizing earlier, it's generating more carbon for those microbes, it's protecting itself, it's generating soil, it's generating health, it's generating photosynthetes.
Yeah, but that it's actually interesting because that happens within the soil because of the microbial community, but there's also, it helps with the plant based off of heat and cold stress, doesn't it?
Yep. Oh, absolutely. So this zone, this creation that these microbes are doing around that root, there's a lot going on in that extracellular polysaccharide. And the other side of that, the beauty is we have now surrounded the plant root with all of these microbes, and they're constantly generating their own communication molecules. They're talking to the plant. They're allowing that plant to get a better idea of what's going on. I think about these microbes out in the soil. They're like scouts. They're out in front of the plant. And if you think about the scale of a plant root compared to a microbe or bacteria, they're considerably different. So these little tiny bacteria, they're much more sensitive to drought temperatures, to drought and temperature, to salinity, to toxins. They have to be more sensitive to that. And when they sense it, many of them that are motile through chemotaxis, they'll move toward what they want to be near and away from what's dangerous. And the beauty is they're then utilizing their own biochemical responses, this biochemical communication with the plant to help protect the plant, depending on what's going on. They may be producing osmolytes or osmoprotectants. If there's moisture loss or the plant is going to be going through osmosis and losing moisture because of drought stress, they'll be producing osmolytes that help protect that plant and help it maintain that moisture. If the temperature gets too high, they'll generate heat shock proteins. As temperature raises, we can have damage, and that's something that we see all the time.
Well, you know, you talk about scouts and basically going out there and letting the plant know basically what is coming. We sometimes talk about that. An example I kind of give is a leaf extract analysis or a plant sap analysis. We talk about by looking at the new leaf and the old leaf, we can have a 2-week period window where we can see basically
What's on the
into the
horizon.
future.
Yeah.
Exactly. So we can see forward. And that's really, this is another tool
Mhm.
that if we don't have that tool, the plant, as we talked about, is there to fend for itself.
Yep.
It can't move.
Nope.
Now it has a communication to, as we say, see what's on the horizon, prepare itself for what is up and coming.
And it goes beyond that. I mean, this communication is incredible. There is so much back and forth going on between the microbe and the plant, and the plant and the microbe. And the plant is purposefully, as we talked about, like Dr. White's talked about, rhizophagy, purposefully generating and cloning those protoplasts of the organisms that it wants and needs. So depending on what's going on, it will upregulate, downregulate, it'll change those species, it'll change what's going on. So one universal that generally goes on when you have any sort of stress is you need energy. As we kind of mentioned before, any damage that occurs requires to be fixed. Anytime you have to fix something, it takes energy to fix it. So one of the first things that we often see is a plant under drought or salinity stress, or heat or cold or any of those stresses, it needs more phosphorus. So we generally see a phosphate solubilization uptick that allows that plant to grow through that stress environment. We also see shifts and changes in a number of the phytohormones, like we talked about. In that drought or salinity, like we talked about, we're going to see a decrease in some of those growth factors and an increase in ethylene. Ethylene is the senescence molecule that encourages the plant to finish up and go to seed. That's not necessarily a great thing if we're trying to get maximum yield, maximum benefit out of that. So by utilizing these plant growth hormone rhizobacteria in that zone, we see a shift in the cytokinins, which are the cell division molecules. We see that upregulation of abscisic acid, like we talked about. We allow the hormones of that plant to open and close the stomata more regularly. We also see those microbes directly feeding the plant some of the antioxidants they can use, as well as upregulating genes for antioxidant development, so that when those reactive oxygen species occur, the plant can break them down. Osmotic stresses are controlled by the osmolytes the microbes are producing. And that ethylene that I mentioned, that senescence molecule, ACC deaminase producing microbes, they actually break down the precursor of ethylene so the ethylene doesn't get there. It's like the plant can't say, hey, hurry up. It's just that is quieted. They turn the volume down on that. We also see, interestingly enough, significant shifts in the root morphology. And we'll talk a little bit more about that. But through the endolytic acid systems, these growth systems, the plant is but the microbes allowing that plant to grow more root, maintain more root. And as we maintain a larger root area, we're able to access more of what that plant needs. And that EPS is then going to bind those materials together. We also see through volatile organic compounds the microbes are producing, they actually allow the plant to move more sodium from its shoot down to the root and out. Then the microbes, utilizing their EPSs, the biofilm, extracellular polysaccharides, bind that sodium into that structure. I compare it to mortar when you're doing bricks. You're sticking it in there, we're taking our individual soil particles, we're gluing them together, and we're binding that sodium or heavy metal, because we see the same thing happens with heavy metals when the microbes and plants are exposed to it and they need to get rid of it. So we see this wide shift of differences that are going on: better nutrients, better growth, decreased senescence, and allowing that plant to move more of what it doesn't need, that excessive material, down and out, and then bind it up. And it's kind of fun, this idea of induced systemic tolerance. What this means is basically you don't even have to have the microbes touching the plant. When they're directly touching the plant, that's one thing, but they don't have to be. Like we talked about the idea of the scout microbes that are kind of out there in the environment a little further away. They're constantly talking. They're constantly generating these these molecules, which are known as volatile organic compounds or volatile organic carbons, they're sending those out to talk to each other and let each other know what's going on. But the plant picks up on those as well. So by simply having those microbes out in the environment, the plant is able to listen to what those microbes are saying. And if the microbes are saying, man, it's getting dry down here. Oh man, there's a lot of salt in this area. Plant knows that it doesn't want to grow in that area, or it's going to upregulate some of the enzymes, some of the molecules that are associated with protecting it from that stressor.
Yeah, you know, and so often we see this out in agriculture. You know, I was in Kansas a few years ago talking with a sod farmer, and it was interesting. Conventional sod farmer, a lot of use of conventional fertilizer.
Mhm.
And he said, I'm seeing more salt grass. And, you know, that makes sense. Irrigated water, poor water quality, higher sodium levels, higher salt levels within
Check,
his
check,
soil
check,
environment.
check.
Yeah. And so, yeah, we are bringing in plants, again, environment dictates expression,
Mhm.
that are more conducive to growing in that environment. And, you know, one of his questions what do I do? I mean, the salt grass, I can't have this in my bluegrass field. This is, you know, seed, obviously. And so we talked about, first off, stop, you know, as we tell growers all the time, if we have a way to stop what we're doing to add to the problem,
Mhm.
we need to do that and let the microbes go to work on it
Yeah.
based off of our conversation, because, you know, they're very good at remediating these types of
Mhm.
things in the soil environment that we have created.
Yep.
But if we continue doing the same thing that we've always done in the past, they can never get ahead.
Mhm.
We're just kind of maintaining a level. So a lot of times as I talk with growers is, let's identify where it's coming from. If it's coming from our water, well, there's not a whole lot we can do there. So we try and do the best we can based off of nutrition and
A
biology.
little bit of humate, some
And-
carbon. Mhm.
Exactly, in order to kind of modify that and just make it so it's not as susceptible. But, you know, so those are a lot of times I think, but a lot of times this is just environmental conditions. You know, I can remember a peach grower in Palisade, Colorado. We used to call it Lake Culbertson because it was a salt flat,
Yeah.
basically. And numerous times he had tried to plant peaches in
Mhm.
that environment and nothing ever wanted to
Or
grow.
anything.
Or anything. No
Yeah.
weeds even grew
Oh
in this
boy.
environment.
Crusted over and
Crust-
just a mess.
Yeah. And so, you know, a lot of what we did is we came in and we broke up that soil. A lot of
Mhm.
times we say iron in the soil is a good thing.
Yep.
This is one of those situations where we had to open that out, get oxygen
Mhm.
so that
Let it
microbial
breathe.
community could have an environment. They needed oxygen, they needed water. And so we put them on a drip irrigation system. I remember the grower calling me and he was so excited because we weren't supposed to put trees in until the next spring.
Mhm.
And he called me and was all excited because he had weeds growing along his drip system. He'd never seen anything else grow there. But it was calcium, it was carbon, it was Spectrum DS,
Mhm.
basically designed for drought stress, high sodium soils,
Yeah.
was placed into that environment. Then we gave a water. Now we have an active growing weed species, which is a great thing because they're designed to be there. They're help mining that out of that environment. We talked about that. Leave them grow, don't kill them. Let them do their job. What we did is we did remove those
Don't let
after
them go
the
to
end of the
seed.
season.
Yeah.
Yeah, we didn't let them go to seed, but we
Mhm.
removed them, cut the plants back, removed all that material. So we weren't just putting it back into the soil environment. And next year we came back and planted peach trees in that area. And it was about a 5-acre block that he ended up doing, if I
Wow.
remember correctly. And when you start to think about that, significant on ROI of ground that you have that before was not usable ground and now is highly productive ground, which is pretty amazing. And the fun part about this story is actually it was during the time when hemp was such a big thing in Colorado.
Mhm.
So in the same area, he had two things going. He had his new peach trees that he
Yeah.
planted. In between the rows of the peach trees, we planted hemp. And he actually had a dual crop growing there. He made a little money. Sometimes we used to do watermelons. He found hemp was a little more profitable for him that year.
Hemp can
And
be profitable too.
yeah, and yeah, he had a good, incredible crop, incredible crop. But a lot of it comes down to we have to change that soil
We do.
environment.
Yeah.
We have to shift from what it was, and there's a lot of ways that not only are the microbes able to shift that over time, but we also have to have those microbial communities around that plant just to balance out some of those stressors.
Yes.
A lot of times, if we can manage those stressors, the plant actually can actively grow.
Mhm.
And that's really what was going on here. And, you know, it's amazing when you start to build that soil structure and you start to build that foundation of those excess cellular polysaccharides that bind those particles. They also give you water holding
Yes, they
capability in
do.
that same thing. And we start to create a situation that is more conducive to actively growing plants.
Well, exactly. And when we're thinking about heat and drought and salt, the reality is most of these all kind of revolve around water.
Water.
Pretty much everything in agriculture, and the most direct indicator of relative crop yield is directly connected to water. If a plant is overwatered or underwatered, it directly impacts yield. So what has the NRCS been saying for a long time? What do we need to do?
27,000 gallons of water for 1% organic matter.
Absolutely.
And, you know, so often I talk to growers, and when we start to go down this, whatever we want to, biological pathway, regenerative pathway, whatever term we want to refer to it as in agriculture, but we have to balance our nutrition. We have to get more organic matter.
Carbon
But
cropping. Let's call it that.
that's what it is. But when we think about that, is everything that we've talked about up to this point based off of photosynthesis,
Mhm.
based off of root exudates, based off of all of these types of things are critical of how fast we build organic matter within that soil environment. So if we have this biological community within that soil environment helping to support that plant through all these environmental stresses that we put out there, we're only going to increase overall soil health and organic matter and soil structure and all the nutrient availability and all these things we want to do. What's really amazing about this, if we think about it, if we want to build soil health, what is the quickest way to do that? Obviously, roots, an actively growing root in that soil. And not only an actively growing root, but if we start to think about root morphology and how that grows and how much quicker we can have contact with that soil environment, put more carbon, more exudates in there, is really critical. I mean, Dr. White talks a little bit about that on root morphology and root structure.
And it's you can't really separate the two. I mean, they constantly go together, and you oftentimes compare the idea of the roots in a conventional or a salt-based fertility program where we're force feeding and piggybacking in, like a freight system, versus our commuter system. It's the individual stops. We want as many stops as we can all along that system. And that all means that we have more access to more nutrition. We're accessing more of the soil. When we pull up or we dig up a plant and we see that dreadlock, that massive bushy material, the massive roots, with all that soil hanging onto it, we know that that plant is going to have better access to moisture, like we've been talking about. It's going to be depositing more carbon in that soil. It's creating soil as it goes. And the research has been showing up to 50% of the carbon can be dead microbial biomass. So those microbes are critical for building our carbon. And they're also critical for helping shift and change that plant's growth patterns. They can literally change how the plant's roots are growing. Like we talked about, again, back to that idea of, the microbes knowing where some of this material is, they're going to help that plant grow in the direction that it needs to grow to access that nutrition. They know where it is. According to Grover et al., 2021, many PGPR, by producing phytohormones, volatile organic compounds, and secondary metabolites, play an important role in influencing the root architecture and growth, resulting in increased surface area for nutrient exchange and other rhizosphere effects. Thus, PGPR capable of modulating root traits can play an important role in agricultural sustainability. That's what the research is saying, and that's what we see day in and day out, is the microbes down there shifting and changing that soil environment, creating that rhizosheath around the root that is completely different from the bulk soil around it. It is this insulated zone. It's a home for the microbes and root.
Well, and as Dr. White's research is showing, those fine root hairs, when I dig up, for an example, like a corn plant, or I dig up out in a wheat field and I see that huge, great root structure, but I see all those fine root hairs coming off of that. All of those are created to spit basically microbial community back out into that soil environment. So when you see all those fine root hairs, you see, okay, biological activity is functioning at a very high level here. The plant is functioning a little bit more as designed. And that's where I get really excited about it. I can talk about an example that we did in a garbanzo field and in the Palouse. And, it was kind of interesting because we went out and, pretty much the way it is any input you add to garbanzos, you never get a return, but that's pretty much the standard practice. So we did a biological inoculum. In this case, it was OP8. And I went out there after planting, after the garbs had come up, and I came out and met with the grower and I said, "Are you seeing any difference?" And he goes, "No, I'm really not seeing any
Visually, you
difference at
couldn't
all."
see much.
You
yeah.
couldn't,
A little bit.
a little bit.
Yeah.
But we had it all flagged out. These were 10-acre trials, fairly large trials. So I said, "Well, let's go down and dig." So we went out and threw a shovel up in the air in the control and dug up a plant. And then we did the same thing on the biologically inoculated side. And the root mass and root structure was significantly different from one to the other. I mean, almost 3 times as large a root
Wow.
mass on the treated versus the non-treated with this biological community. And that's what we're talking about.
And you're thinking nutrients. Yeah.
This root morphology.
Uh-huh.
And I'm looking at the amount of nodules that are on the treated versus the non-treated, and nitrogen fixation and all that goes along with that. And he's looking at these roots are going to get me through the drought stress period, which is going to give me higher yield. I'm going to fill those pods. Usually what happens is you get the pod set, but you can't fill them because you run out of moisture.
Yep.
And we talked about that. Moisture is key to overall plant health. I mean, without moisture, the plants don't grow. And there's not a lot we can do about that. There's some things, but at some point it's going to take over. But with this basically 2 times the amount of root mass on this plant, we're mining more soil for that moisture. The amazing part about that is we talk about row closure in a lot of crops. And one of the things that we immediately noticed was the plants that were under the biological were thick and full, and there was no ground showing.
Shaded,
which retained moisture within that soil environment. Cooler soil temperatures, all better environment for microbial communities and overall plant health. And we ended up doing much better on the yield. It was pretty exciting.
It's almost like Mother Nature is smart and knows what she's doing when we get out of her way.
Well, sometimes she kind of designed that system, didn't she?
Weird, I know, and it's so cool, and it's fun going out with growers and seeing that and digging it up. When you first go out with a grower and they're not used to the smell, they're not used to seeing the different insects, the different species that you can have out there. It's so fun seeing life start to flourish in some of these environments. Okay, sorry. Anywho, back to abiotic stress. So we wanted to focus a little bit on, we talked a little bit about salinity, we talked a little bit about moisture. Now we want to talk a little bit about heat and drought. Again, a lot of this boils down to moisture. When we talk about holding on to moisture, we need to have the plant needs to maximize the amount that it can store and hold on to. Part of that is changing its root architecture, changing those, creating that rhizosheath, holding on to more, depositing more carbon. But also part of that is the stomata, opening and closing of the stomata. That is directly related to the microbes down in the soil. The microbes down in the soil are communicating with the plant. And like we talked about, they tell the plant when it's starting to dry out. Research has shown that the plant can more quickly open and close its stomata with the presence of plant growth promoting rhizobacteria than without. So when the microbes sense a dry environment coming along, they, with their hormones, communication molecules, allow that plant to close its stomata faster, holding on to more moisture. We also see an upregulation of potassium mobilizing bacteria because we know that that plant is going to need potassium because that is necessary for opening and closing the stomata. So this heat is a little bit different. Heat stress and drought stress, it all kind of binds together. You start with drought when you run out of water, but we're also seeing a lot of environments where we just have plain heat damage. And heat damage is hard because we start to have what's known as protein denaturation. We're breaking down the structure of proteins, and some of those intracellular molecules, some of those intracellular organelles and structures, they are physically damaged by the heat and they break apart. The problem with it is once those proteins break down, they're done. They don't work like they were supposed to, meaning the plant has to clean them up. What did I say earlier? Anytime we have to clean something up, takes a lot of energy. So plant has to break that material down and then has to rebuild that material, which takes more energy. So we want to avoid that heat stress. We want to avoid that as much as possible. There are specific molecules that can be produced known as heat shock proteins, and those purposefully kind of encapsulate and protect some of those proteins so they no longer break down. And guess what? Microbes can help with that. They upregulate the generation of them by the plant, so it doesn't have to break down those materials. It helps protect them before that damage is done, as well as the microbes are generating and feeding some of those to the plants themselves. So we see a shift and a change in both the hormones. We see a shift and change in the delay in senescence, so the ethylene levels. The volatile organic compounds are allowing that plants to grow through it. Research Shafiq et al. 2022. There's growing interest among researchers in the use of beneficial microorganisms. Intricate and highly complex interactions between plants and microbes result in alleviation of heat stress. And some of these things, we don't fully understand all the communication that's going on. We can measure a lot of it, but we don't fully understand it. But we're seeing these shifts and changes in stomata conductance, opening and closing, moisture holding capacity, photosynthetic efficiency, respiration rate, as well as thermotolerance of these plants. And part of that has to do directly with nutrition.
Yeah, and so much of that, we talk about the importance of this, and heat stress doesn't only happen basically in a dry land scenario. We can have heat stress, not drought stress, but heat stress in an irrigated field.
Sure.
A prime example of that is 2 years ago in Kansas, during the time when the plant corn was pollinating, and this was irrigated
Sensitive
corn,
time.
sensitive time. There were thousands of acres that did not pollinate correctly because the pollen tubes had collapsed because of a heat stress that came on right at that time. I spent 2 days driving around with a field scout out there looking at acres that were being cut as silage, because basically you had so many skips, you didn't have full kernels. You did not have a full ear of corn.
Looking at the cobs, it looked like about 50%.
50%. As where the grower that was working with this biological, that had been biological for probably 15 years now. I mean, his corn all had full ears, had it pollinated properly, and it was amazing. And this field scout came down to the idea of the stress reduction was a huge part due to the biological agriculture practices that he was using, but also due to the calcium that he had in the plant. Early calcium, because those pollen tubes did not collapse under that severe heat. Exactly. When you start to look at that, and it's kind of cool because some of the research that we're looking at like at biomakers, and I just did a test the other day, and it was kind of interesting. I pulled this out, that the calcium transport in the treated versus the control had 40%. The treated had 60%.
On a scale of 0 to 100.
On a
20-point increase.
20-point increase on calcium transport of that calcium for that plant. I mean, that's pretty amazing. When you start to think about that, for an example, when we start to think about that corn and that pollen tube, calcium is not mobile. You are not going to come in with foliars and build that calcium. This has to start at the point, as we always talk about, at the point of germination.
Those babies.
That's where this all starts at. And it's just kind of amazing when you start to think about how everything is interconnected. Bruce used to talk about that. About all these things these microbial communities doing, not only based off a reduction of plant stress, but also of nutrient availability. And when we look at all of these, how they tie together in overall plant health and yield, it goes well beyond just one or two things. I mean, it is just, as Dr. Hatfield talks about, it's a stair-step climb.
It is.
Whether you're building soil health, whether you're building plant health, whether you're building microbial communities within that soil environment, they're all stair-step climbs that we kind of have to go through there. So, with that being said, one of the things that we want to talk about, how do you get that stair-step climb? Get those friends out there. Get these inoculums into that environment. Get an actively growing root. Do cover cropping when you can do that.
Carbon.
Don't forget, in your cover crop, seed treatment, the exact same things happen there. Your foliar nutrition is huge. It has to be a part of this whole system. We have to do everything we can in order to enhance microbial diversity within that environment, which is really kind of interesting, because in the same trial that we looked at basically seeing the calcium transport, one of the things that we noticed is the control versus the inoculated. The inoculated had basically a 41% increase in functional biodiversity.
What's functional biodiversity? How would you describe, what does that mean?
You know, I guess my example of functional biodiversity would be if you open up a toolbox and you need a 3/8 wrench, but you have a whole toolbox full of 9/16. That's all you have in your toolbox. You have 156 9/16 wrenches, but you can't do anything with that 3-inch.
Nope.
Where you look at the biodiversity, you have screwdrivers, you have metric, you have standard, you have sockets, you have end wrenches, you have an impact driver, you have a torque wrench, you have them all out there. So if you need that tool, if the plant needs that tool, he can send that communication down, and that tool now can function as designed. I mean, that's really what biodiversity comes into, and that's what we talk about all the time, is we want all of these microbial communities within that soil environment. So if they are needed, they have the opportunity to function as designed.
Well, like that Montana State University research that we were doing, where we were looking at just PGPR, and by inoculating with PGPR, they also saw an increase in fungal communities. As we start this process, it's incredible. We can put out a few of the stepping stones, and then Mother Nature, she craves and thrives on building these systems back up if we can get out of her way.
Well, and these are plant growth promoting rhizobacteria. They're designed to increase plant health. As we increase plant health, we increase exudates. We increase carbon. As we build soil structure and increase carbon, we create an environment. We create habitat for all of these other species, all these other fungal communities, all these other bacteria to be there. We've created an environment that is conducive to growing them all. And that's really what we're trying to do. That's how we truly build soil health. Well, and I guess with that,
I had fun.
I had a great time. So thanks, everybody. Thanks for listening to us. We hope you got something out of it. Again, I guess I would want to say that this is the Natural Growing Through Biology podcast. I'm Dennis.
I'm Steve.
And we hope to talk to you soon.
Thank you.